What changes first in evolution? An organism's genes or its traits? Cavefish help us to answer this question. When blind Mexican cavefish were first discovered, they appeared so different from other fishes that they were listed as an entirely new species, only for subsequent research to establish that normal-sided versions of the same species are widespread. Moreover, the blind and sighted fish interbreed and produce fertile young. At least 30 cavefish populations are now known, mostly eyeless and with reduced pigmentation, but also exhibiting changes in bone structure, metabolism, and foraging. That so many cave populations have independently evolved the same characters has led researchers to wonder whether plasticity might have played a role in their adaptation to the cave environment. It's well known that development is sensitive to environmental cues, a phenomenon called phenotypic plasticity. For instance, plants grow different leaves when grown in the light or the shade, while hares and foxes have distinct summer and winter coats. Perhaps fish develop differently in a cave. If so, does that plasticity help to explain the convergent evolution of cavefish? When researchers raised river fish in darkness, the fish strikingly resembled their blind cavefish cousins. The findings hinted that plasticity could have played a guiding role in the evolution of cavefish adaptations. But for plasticity to shape genetic evolution, any initial phenotypic modifications must be followed by and partly determine genetic change through natural selection. Evidence is provided by an experiment that subjected river fish to a chemical that mimicked the stressful physiological effects of the cave. Surface fish reared in the presence of the chemical exhibited unusually large variation in eye size. The researchers reasoned that if the phenotypic variation generated by this stress had played a critical role in the evolution of eye loss, it would be observed in surface fish but not cavefish. That's because the cavefish will have undergone selection for reduced eye size that will have eradicated much of this variation, a process known as genetic assimilation. Sure enough, treatment with the chemical revealed extensive phenotypic variation in eye size in river fish, but not cavefish. Moreover, when researchers bred new generations from stressed surface fish with small eyes, they found that eye size responded to selection and got smaller. Apparently, plasticity may have played a critical role in cavefish evolution by exposing heritable variation in eye size and other traits to selection. The parallel evolution of cavefish makes sense once it's recognized that cavefish traits do not evolve independently, but are connected through interacting developmental mechanisms. Plasticity may help the population survive the challenge of finding oneself in a cave until natural selection devises more robust adaptations. However, plasticity does more than buy time for natural selection. It helps determine the phenotype distribution, shaping the correlated variation that is exposed to selection and thereby influencing the rate and direction of evolution. Sometimes it seems plasticity really does lead and genes follow. Want to explore this topic further? Check out Evolution Evolving now.